Greetings everyone, and today we are going to discuss about classification of enzymes. Classification of enzyme can be classified on the basis of the reactant and substrate. On the basis of substrates, there are several enzymes such as lipase, protease, amylase, and nuclease. These are enzymes which basically are classified on the basis of their substrate. For example, we are having lactase. Lactase basically catalyzes lactose. Protease catalyzes protein. Nuclease catalyzes nucleic acid. RNase catalyzes RNA, and ATPase catalyzes ATP, and so on. There are different types. Now we are majorly discuss about reactants, enzymes classification on the basis of the reactants reaction on the basis of the reaction. So first enzyme includes oxidoreductase. Oxidoreductase EC1 is basically the enzyme which is involved in oxidation and reduction processes by transfer of electrons from one molecule to another, or removal and addition of hydrogen molecule, uh, addition and removal of water, oops, removal, addition and removal of electrons, addition and removal of protons, as well as addition and removal of oxygen. It includes lipoxidases, dehydrogenases, glucose oxidases. Transferase basically catalyzes the reaction by transferring one group of atoms from one molecule to another. For example, we are having hexokinase, aminotransferase. No, we are not having hexokinase, we are only having aminotransferase and transaminase, which transfers one group of atoms from one molecule to another molecule. And apart from this, we're having hydrolase. Hydrolase enzyme which undergo the process of hydrolysis in order to uh, perform the cleavage of substrates by water, and that includes lactase, protease, trypsin. All of our digestive enzymes. Lyase involves all of the enzymes which catalyze the addition of groups to bond double bonds, or the formation of double bonds via the removal of groups in such a manner that they do the addition of groups to double bonds, or the formation of double bond via the removal of groups without hydrolysis, which includes pectate lysis, decarboxylase, hydratase. Isomerase is a type of enzyme which simply uh, catalyzes the intramolecular arrangement of atoms of molecules. It catalyzes the intramolecular arrangement of atoms of molecules, such as we are having phosphohexose. Phosphohexose basically converts the glucose 6 phosphate into fructose 6 phosphate, which is isomer of glucose. In that case, this is a function of isomerase. Similarly, we are having topoisomerase, glucoisomerase, and when there's ligase basically is an enzyme which is involved in catalyzing the joining of two molecules to form a new bond. For example, RNA polymerase, DNA polymerase, glutathione synthase, amino acid tRNA synthetase are all enzymes that are involved in ligase. Translocase are those enzymes which catalyze the movement of ions of molecules across membranes. That are catalyze the movement of ions or molecules across membranes, or their separation within membranes. For example, we are having ATP synthase, ascorbate, parareductase, ubiquinone reductase. These are all enzymes which are involved catalyzing the movement of ions of molecules across membranes. And hexokinase is an enzyme, as well as we having alcohol dehydrogenase, which converts alcohol into acetaldehyde through the process of uh, through the process of catalysis, and that is involved in the example of transferase. On the other side, hexokinase having example of, we are having hexokinase inside the isomerase, because hexokinase basically converts hexokinase converts hexokinase converts hydrogen hexokinase converts glucose into glucose 6 phosphate, due to which uh, it is involved in. All right, we've got it. So here isomerase involves example of phosphohexose, whereas transferase includes uh, hexokinase. Transferase includes hexokinase. That's all about today's video. Hopefully we'll meet again in another.